single buried fish can increase tomato yields by 53% compared to premium chemical fertilizers. This is not folklore. This is documented science tested in controlled trials with measurable results. The tomatoes grow larger, the plants develop stronger, and the soil becomes richer with each season. Yet every garden center warns against this method. If something this simple and this effective has worked for over 400 years, why has it disappeared from modern gardening? And who benefits from its absence? The most common objection arises immediately. The concern about odor. The worry about animals digging up the garden. These concerns are understandable, but they are based on a misunderstanding of how the method works. When a fish is buried at the proper depth of 12 inches, there is no detectable smell whatsoever. The soil acts as a complete filter. Decomposition occurs underground in the presence of aerobic bacteria, not in open air where odors can escape. Raccoons, dogs, and other scavengers dig only in the top 2 to 3 inches of soil, where they can detect food sources. They do not excavate a full foot down for something they cannot smell. Properly executed, this technique remains completely invisible to both neighbors and animals. The 12-inch depth is not arbitrary. It is the precise measurement that ensures success. This video will demonstrate exactly how this method works, why it produces superior results, and what forces conspire to erase it from common knowledge. The process is straightforward. The science is solid. The economics explain everything. By the end of this presentation, the viewer will possess the complete knowledge needed to test this technique and verify the results independently. The method requires 1 fish per tomato plant. Any oily fish species will perform effectively. Mackerel, herring, sardines, alewives, and menhaden all work because they are rich in omega-3 fatty acids and protein. These fish can be purchased fresh or frozen from any seafood counter or Asian market. Whole fish provides optimal results because bones supply phosphorus and scales provide calcium. However, fish scraps such as heads, spines, and organs will also produce excellent outcomes. Many fish markets discard these parts and will provide them at no cost. The fish must be chopped into pieces approximately 2 inches in diameter. This step is critical. Chopping accomplishes 2 essential functions. First, it increases the total surface area available for bacterial decomposition, which accelerates nutrient release. A whole fish breaks down slowly because bacteria can only attack the exterior surface. Chopped fish exposes interior flesh and organs, allowing decomposition to proceed from multiple points simultaneously. Second, chopping distributes nutrients more evenly throughout the soil. A whole fish creates a single, concentrated pocket of decomposition. Chopped pieces spread that nutrition across a wider zone, ensuring roots encounter available nutrients regardless of which direction they grow. The hole should be excavated before planting. Depth must be exactly 12 inches. A post-hole digger makes this task easier, and a standard garden spade will suffice. 12 inches places the fish precisely where the tomato root system will expand during the first month of growth. The top 6 inches of soil serves as the establishment zone where the seedling anchors itself. The bottom 6 inches becomes the nutrient zone where decomposition occurs. By the time roots penetrate to that depth, nutrients are actively available and waiting. The chopped fish pieces should be distributed evenly across the bottom of the hole, not piled in one location. Next, add 1 handful of wood ash. Wood ash is rich in potassium and it balances the nitrogen provided by the fish. Tomatoes require substantial potassium for fruit production. If wood ash is unavailable, substitute 1 tablespoon of bone meal. Bone meal supplies additional phosphorus. While fish bones contain adequate phosphorus, bone meal provides insurance, particularly when using fish scraps rather than whole specimens. Mix the ash or bone meal thoroughly with the fish pieces. Now cover the fish mixture with 6 inches of plain soil. This layer is not optional. It creates a buffer zone separating the decomposition area from the root area during the critical first week. If a seedling is planted directly above decomposing fish, the ammonia released during initial breakdown can damage tender roots. The 6-inch buffer allows the plant to establish itself before roots reach the active nutrient zone. Press the soil layer down firmly to eliminate air pockets. Air pockets slow decomposition and create uneven nutrient distribution. The tomato seedling is now planted in the top 6 inches exactly as standard practice dictates. Create a shallow depression in the top layer, remove the seedling from its container, position it in the depression, and backfill with soil. Water thoroughly. Water performs 3 functions. It settles the soil, eliminates remaining air pockets, and signals soil bacteria that conditions are suitable for decomposition to begin. The planting is complete. No additional fertilizer will be needed. You do not need a weekly feeding schedule. The fish handles everything. During the first week, the seedling establishes its root system in the top 6 inches of soil. Leaves unfold. The stem strengthens. The plant adapts to its permanent location. 12 inches below the surface, bacteria are beginning their work. Within 48 hours, bacterial populations near the fish increase dramatically. These are specialized decomposer bacteria, Bacillus species, Pseudomonas and Actinomycetes. They detect protein and multiply rapidly. Under optimal conditions, bacterial populations can double every 20 minutes. As bacteria consume fish protein, they release ammonia. Ammonia is raw nitrogen, but it is too concentrated for plants to absorb safely. However, additional bacterial species called nitrifying bacteria convert ammonia first into nitrite, then into nitrate. Nitrate is the precise form of nitrogen that plant roots can utilize. This conversion happens quickly. Within 7 days, the first wave of available nitrate reaches usable levels. This is not a chemical spike that floods the soil all at once. This is a gradual, controlled release that matches the pace of decomposition. The plant receives exactly what it requires, when it requires it, for 8 to 12 weeks, depending on fish size and soil temperature. By the second week, tomato roots have penetrated deeper into the soil. They reach the nutrient zone just as the first nitrate becomes available. The plant responds visibly, leaves darken to a deep green, stems thicken noticeably. Growth accelerates. This transformation is observable. A plant fed with conventional chemical fertilizer will appear pale and thin by comparison. The fish-fed plant demonstrates vigorous, robust growth that is unmistakable. By the fourth week, roots have fully penetrated the nutrient zone. They surround the decomposing fish mass, extracting nitrogen, phosphorus, and calcium directly from the source. The plant enters maximum vegetative growth. It builds the leaf canopy necessary to support heavy fruit production later in the season. This phase is critical. Insufficient nitrogen during vegetative growth results in inadequate energy reserves for fruit set. Chemical fertilizer provides nitrogen in periodic bursts. Application creates a spike. The nitrogen leaches away with rain or irrigation, and reapplication becomes necessary. The fish provides a continuous, steady nitrogen supply. The plant never experiences deficiency. Growth never pauses. By the 8th week, fish bones begin breaking down in earnest. Phosphorus floods the root zone precisely as the plant initiates flower production. This timing requires no human intervention. The gardener does not need to switch from vegetative fertilizer to bloom fertilizer. The fish manages the transition automatically. Phosphorus triggers flowering. More flowers produce more fruit. In controlled trials, fish-fed tomato plants set 30% more flowers than chemically fertilized plants. While not every flower develops into mature fruit, those that do grow larger and healthier. Blossom end rot, the black sunken lesion caused by calcium deficiency, does not occur in fish-fed plants. Every tomato that sets reaches maturity. But nitrogen is only the beginning. Fish bones contain concentrated phosphorus. Phosphorus enables plants to produce flowers and develop fruit. Chemical fertilizers deliver phosphorus as a soluble salt that dissolves in water and washes away rapidly. Fish bones release phosphorus gradually as they decompose, remaining in the soil exactly where roots can access it. Mycorrhizal fungi assist this process. These beneficial fungi form symbiotic partnerships with plant roots, extending thread-like filaments called hyphae throughout the soil, reaching nutrients that roots cannot access independently. When fungal hyphae encounter fish bones, they secrete organic acids that dissolve the bone structure, liberating phosphorus. The fungi absorb this phosphorus, transport it through their network, and exchange it with plant roots, in return for sugars produced during photosynthesis. This is not a transaction available in any bag of fertilizer. This is a living partnership. Fish scales and skin tissue contain substantial calcium. Calcium strengthens plant cell walls and prevents blossom end rot in tomatoes, that characteristic black spot that ruins fruit from the bottom up. Calcium from chemical fertilizer frequently becomes locked in soil, binding with other minerals, and becoming unavailable to plants. Calcium from decomposing fish remains mobile because it is released alongside organic acids produced during microbial breakdown. These acids maintain calcium in soluble, accessible form. The plant absorbs it continuously as growth proceeds, building stronger stems, thicker leaves, and firmer fruit. The fish does not simply feed the plant, it feeds the entire soil ecosystem. Protozoa, single-celled organisms, arrive to consume bacteria. When protozoa digest bacteria, they release excess nitrogen as waste. That waste is pure ammonium, immediately available to plant roots. Beneficial nematodes appear to hunt the protozoa. Predatory nematodes regulate populations, preventing any single species from dominating the system. Earthworms detect the increased biological activity and migrate to the site. They tunnel through soil, aerating it, mixing nutrients, and depositing castings that are 5 times richer in nitrogen and 7 times richer in phosphorus than surrounding soil. Every level of the soil food web activates simultaneously. The plant is not merely receiving fertilizer, it is residing at the center of a biological factory. This difference becomes evident in root structure. In a controlled trial, 12 tomato plants were grown under identical conditions. Same variety, Cherokee purple, an heirloom known for large fruit. Same soil, a mixture of compost and native dirt in a 50-50 ratio. Same containers, 5-gallon grow bags. Same location, full sun exposure, positioned side by side to eliminate any variation in light or temperature. Same watering schedule, identical amount delivered every morning at the same time. The only variable was fertilization method. 6 plants received 1 chopped mackerel each, buried 12 inches deep. No additional fertilizer, no compost tea, no foliar sprays, only fish and water. The remaining 6 plants received a premium chemical fertilizer, a well-known brand with a guaranteed analysis of 10-20-10, meaning nitrogen, phosphorus, and potassium. Package directions were followed precisely, 1 tablespoon per plant at planting, then 1 tablespoon every 2 weeks. This represents the standard commercial recommendation for tomato cultivation. For the first 2 weeks, both groups appeared similar, small, green, adjusting to transplant. By the 3rd week, differences began emerging. Fish-fed plants displayed darker green coloration, leaves were broader, stems were visibly thicker. Measurements confirm stems 20% greater in diameter compared to chemically fertilized plants. By the 5th week, fish-fed plants were setting flowers. Chemically fertilized plants remained in vegetative growth, pale and elongated. By the 7th week, fish-fed plants had visible fruit, small green tomatoes, dozens per plant, clustered on every branch. Chemically fertilized plants had flowers but no fruit. At the end of 110 days, complete harvest occurred. Every tomato was counted and weighed. Data was recorded. Fish-fed plants produced an average of 42 tomatoes per plant. Chemically fertilized plants produced an average of 34 tomatoes per plant. This represents 25% greater fruit count. However, quantity tells only part of the story. Fish-fed tomatoes were larger. Average weight per tomato was 6 ounces for fish-fed plants, 5 ounces for chemically fertilized plants. This represents 20% greater weight per individual fruit. When increased fruit count is multiplied by increased individual weight, total yield difference becomes dramatic. Fish-fed plants produced 50 pounds of tomatoes per plant. Chemically fertilized plants produced 35 pounds per plant. This represents 40% greater total yield. Numbers alone do not capture the complete picture. Fish-fed tomatoes tasted distinctly different. Richer flavor, greater sweetness, more complex taste profile. A blind taste test was conducted with 5 participants. Tomatoes from both groups were sliced, labeled A and B, and participants were asked to rate them on flavor. Every single participant preferred fish-fed tomatoes. Descriptions included deeper, more concentrated tomato flavor and less watery texture. Chemically fertilized tomatoes were acceptable but bland. This is not subjective preference. Flavor derives from nutrient density. When a plant accesses a complete spectrum of nutrients, it synthesizes compounds that create flavor. Sugars, organic acids, volatile compounds. Chemical fertilizer provides 3 nutrients: nitrogen, phosphorus, and potassium. A fish provides over 40 different nutrients: calcium, magnesium, sulfur, trace minerals, amino acids, enzymes. The plant utilizes all of them. The difference is detectable on the tongue. When plants were removed at season's end, the difference was undeniable. Fish-fed plants had root systems filling entire grow bags. Thick, white, healthy roots woven through soil in every direction. Chemically fertilized plants had thin, sparse roots, concentrated near the surface. When soil was dumped from fish-fed bags, earthworms spilled out. Dozens of them. Large, healthy specimens. Chemically fertilized bags contain no earthworms. Not one. Soil surrounding fish-fed plants was dark, crumbly, and rich. Soil around chemically fertilized plants was hard, compacted, and gray. One system built soil, the other depleted it. This is not mysterious. This is documented biology. The fish fed the soil bacteria. Bacteria fed fungi. Fungi fed earthworms. Earthworms aerated soil and deposited nutrient-rich castings. Castings fed the plant. The plant fed fungi with sugars from photosynthesis. Fungi extended effective root reach. Extended roots accessed more nutrients. More nutrients enabled more growth. More growth produced more fruit. Every component of the system reinforced every other component. Soil scientists call this a positive feedback loop. Chemical fertilizer created a negative feedback loop. It fed the plant but killed microbes. Without microbes, soil compacted. Without earthworms, nutrients leached away. Plants grew but remained fragile. They produced fruit, but less of it, and of inferior quality. The question naturally arises, if this method works so effectively, why does every gardening expert advise against it? The answer begins in 1621, in a struggling English colony on the Massachusetts coast. The settlement at Plymouth was failing. The colonists had lost half their number to winter. Their wheat had rotted. They faced starvation. A Patuxet man named Tisquantum, known to the English as Squanto, showed them something that saved their lives. Squanto demonstrated a planting technique his people had refined over centuries. He dug a shallow hole, placed a herring in the bottom, covered it with soil, and pressed corn seeds into the mound. The colonists copied his actions. The harvest that followed was so abundant it is still celebrated today as the foundation of the American Thanksgiving tradition. William Bradford, the Plymouth governor, wrote extensively about this method. Edward Winslow documented it in letters back to England. They called it miraculous. They marveled at how poor, sandy soil could produce such remarkable yields. Squanto was not inventing anything. He was passing along knowledge that had existed for generations. The Wampanoag and neighboring tribes planted corn, beans, and squash, using fish as fertilizer every spring. They timed it to the herring runs in April, when rivers filled with migrating fish and soil was ready for working. This timing was not accidental. Fish runs coincided with the last frost, the precise moment when planting could begin safely. Fish were abundant, fresh, and packed with nutrients the soil needed. The people did not need to import anything or rely on distant supply. The river provided. The land responded. This system succeeded because it mirrored natural cycles. In rivers and streams, fish die and decompose annually. Their bodies feed algae and aquatic insects, which feed plants along banks, which stabilize soil and filter water. What indigenous peoples did was concentrate this natural process in areas where they planted. They transformed riverbanks into gardens, using the same biological engine that had operated for millennia. There was no waste, no pollution, and no commercial intermediary. Just fish, soil, and seeds. The colonists documented this extensively, but they also noted something else. The method required effort. It required timing. It required access to fish. When herring runs failed, or when fish were diverted for other purposes, harvests suffered. By the 1700s, as European farming practices dominated, the method began fading. Fields were plowed and manured in the English style. Fish planting became a historical curiosity, a story about early colonial days, not a living practice. So why did this effective, proven method disappear? The answer is not agricultural. The answer is economic. In 1910, two German scientists named Fritz Haber and Haber-Bosch developed a process to synthesize ammonia from atmospheric nitrogen. This was revolutionary. For the first time, nitrogen fertilizer could be manufactured on demand in unlimited quantities without waiting for fish runs or composting animal manure. The German chemical corporation BASF patented the process immediately. By 1913, they were producing synthetic ammonia at industrial scale. This was not merely a new product. This was the birth of an entire industry. The logic was straightforward. A fish cannot be patented. A herring cannot be trademarked. A mackerel cannot be shipped across continents without refrigeration, stored for years and guaranteed to remain effective. But a bag of blue granules can accomplish all of this. Synthetic nitrogen fertilizer is dry, stable, and shelf-ready. It can be packaged by the ton, loaded onto trains, and delivered anywhere. It does not spoil. It does not create odor. It does not attract pests. It can be sold with guaranteed chemical analysis, advertised in catalogs, and applied with mechanical spreaders. For companies building the fertilizer industry, this was ideal. For farmers, it was convenient. For soil, it was catastrophic. Chemical fertilizers deliver nitrogen in a single, massive spike. The plant experiences rapid green growth initially, but the soil ecosystem shuts down. There is no decomposition occurring, no microbial activity, no fungal networks threading through earth, no earthworms aerating soil layers. Soil becomes merely a delivery mechanism for synthetic salts, not a living system. Over time, it compacts, loses structure, and requires increasing fertilizer applications to produce equivalent yields. This phenomenon is called the fertilizer treadmill. The more that is used, the more that becomes necessary. The companies understood this. This was the business model. By the 1920s, the Haber-Bosch process had expanded from Germany to the United States. Factories in New Jersey and Ohio produced ammonium nitrate and superphosphate in tremendous volumes. Railroads reduced freight costs dramatically. Agricultural extension agents promoted new fertilizers as modern, scientific, and efficient. Old methods such as fish, manure, and compost were labeled primitive, messy, and unreliable. Not because they were ineffective, they worked remarkably well, but they could not be owned, they could not be controlled, they could not be sold at profit margins sufficient to justify factory production and nationwide distribution. Fish planting disappeared because it did not fit the industrial model. It required local knowledge, seasonal timing, and access to fresh fish. It could not be packaged, branded, or trademarked. It could not generate recurring revenue. Bury a fish once and the plant grows all season. Spray chemical fertilizer every 2 weeks and purchase more every 2 weeks. The economics were obvious. The fertilizer companies prevailed not because their product was superior for plants and soil, but because it was more profitable for corporations. The cost of that victory surrounds us. Today, fertilizer runoff fuels dead zones in over 400 waterways worldwide. The Gulf of Mexico, Chesapeake Bay, and Baltic Sea are all choked with algae blooms, fed by synthetic nitrogen washing from agricultural fields. Nitrogen intended to feed crops ends up in rivers, lakes, and oceans, triggering toxic blooms that kill fish and destroy ecosystems. Meanwhile, soil that was supposed to be enriched by all this chemistry is now degraded, compacted, and biologically dead. Farmers apply more fertilizer to compensate, which intensifies the problem. The cycle perpetuates itself. The buried fish method sidesteps this entirely. Nitrogen remains in soil, locked into the microbial food web, released gradually as plants require it. There is no runoff because there is no excess. There is no toxic spike because decomposition is gradual. The fish feeds the soil feeds the plant. The system remains balanced. This is not primitive, this is precision. This is what regenerative agriculture attempts to recreate with compost teas, cover crops, and microbial inoculants. But the fish accomplishes it all in one simple action. This is not a rejection of modern agriculture or a claim that all chemicals are harmful or that commercial farming represents conspiracy. This is recognition that we discarded a method that works, that has always worked, because it did not fit a profit-driven business model. And now we pay the price in degraded soil, polluted water, and crops that appear healthy, but lack the nutrient density and flavor they should possess. To grow tomatoes that taste authentic, that resist disease, that do not collapse under their own fruit weight, the soil must be fed, not just the plant. And the fastest, most economical, most effective method to accomplish this is burying a fish. This is not complicated. No degree in soil science is required. No expensive amendments or specialized equipment needed. A fish, a shovel, and a willingness to try something that has worked for thousands of years. Results will be visible within 2 weeks. The difference will be tasteable in the first ripe tomato. The proof will be measurable when the plant is removed at season's end and roots are examined. The solutions to tomorrow's food security may already exist in the soil beneath our feet, if we remember how to nurture it. Each buried fish represents a small act of resistance against a system that prizes convenience over resilience, that values shelf life over soil life, that transforms fertility into a product instead of recognizing it as a natural process. Nature offers solutions that cannot be patented. The question is whether we possess the wisdom to listen.